Welcome to this explainer. Today, we are looking at a moment in science that is, quite frankly, going to change the world as we know it. We've reached a point where artificial intelligence has crossed an absolutely massive, unprecedented threshold in biology. We aren't just talking about AI writing college essays or generating funny images anymore. No, we're talking about AI writing the literal code of life itself.
So think about this for a second. What if AI could design entirely new life forms from scratch? I know, it sounds exactly like the plot of a sci-fi movie, but it is happening literally right now in laboratories. And as we're going to explore today, the answer to that question is the ultimate double-edged sword. On one hand, it offers a miraculous potential cure to some of our most terrifying medical challenges, but on the other, well, it introduces a biosecurity threat so severe that experts are scrambling to figure out how to contain it.
To really understand why scientists are pushing this boundary in the first place, we have to set the stakes. We are currently staring down a massive, growing medical crisis: antibiotic-resistant bacteria. You've probably heard of these superbugs. They're mutating way faster than our traditional medicines can keep up, and they're already killing hundreds of thousands of people globally every single year. If we don't find a radical new solution, a simple scrape on your knee or a routine medical procedure could once again become lethal. We absolutely need a weapon that can evolve and adapt just as fast, if not faster, than the bacteria themselves.
Well, to find that adaptive solution, researchers looked straight to AI. You are likely familiar with ChatGPT, right? It learns human language to predict and generate text. But scientists at Stanford University and the Arc Institute took that exact same foundational AI architecture and built what they called genome language models. They named them EVO1 and EVO2. Instead of reading books or internet articles, EVO was trained on raw genetic information from millions of living organisms. It basically learned the complex syntax and grammar of DNA, allowing it to generate single nucleotide biological sequences completely from scratch. Now, I cannot overstate how incredibly difficult this task is. We're talking about genome-scale design at a single nucleotide resolution. It's kind of like writing millions of lines of computer code where a single typo crashes the entire operating system. DNA isn't just a random string of letters, it has overlapping reading frames and complex structural dependencies. The researchers identified a massive risk here. If even a single nucleotide base pair is inserted in the wrong place, it can completely destroy the entire organism. It is a biological tightrope walk that requires just absolute extreme precision.
But here is the mind-blowing part. They actually pulled it off. 16. That is the number of biologically viable, entirely new bacteriophage viruses that this artificial intelligence successfully generated. Let that sink in for a moment. An AI model envisioned the DNA of a virus that has never existed in nature. Scientists synthesized that DNA in a physical lab, and it actually worked.
So, how does this actually translate to tackling that antibiotic resistance crisis? Well, the researchers modeled these new viruses after a natural bacteriophage called phage. Basically, it's a virus that specifically hunts and kills bacteria. EVO generated hundreds of thousands of candidates, and of those, 16 synthetic phages were biologically viable. They successfully bursted open and replicated within E. coli bacteria. And get this, some of these AI-generated viruses achieved even faster bacterial killing rates than the natural ones. Even better, when they used a cocktail of these generated phages, it successfully overcame bacterial resistance in just 1 to 5 passages or generations of the bacteria. It's an incredible breakthrough. It means we could theoretically use AI to continuously generate new therapies that outpace those rapidly evolving superbugs.
Okay, but we need to take a deliberate pivot here. This research is the textbook definition of high risk, high reward. Because while the ability to genetically engineer entirely new viruses is undeniably a medical miracle, that exact same technological capability has sparked intense, high-level biosecurity concerns. Think about it. The underlying technology doesn't inherently know the difference between a virus that cures and a powerful bioweapon that kills.
And this brings us to a terrifying juxtaposition. On one hand, we have the incredible capabilities of genome foundation models to edit genomes and design proteins. But on the other hand, we face the reality of jailbreaks. If you follow the AI space, you know a jailbreak happens when someone uses clever prompts to bypass a system's safety rules, tricking the AI into giving out restricted, dangerous info. But if an open-source biological AI is jailbroken, the consequences leave the digital realm and enter the physical world.
Let's unpack the specific threats identified by biosecurity experts. With unrestricted access, malicious actors could repurpose these high-reward tools for absolute devastation. They could ask the AI to design novel toxic proteins, or genetically modify existing viruses to make them more lethal, or help them evade vaccines. There are also huge risks of extracting sensitive patient data from medical models. And it doesn't stop at biology, it extends to chemical threats too. We're talking about generating the exact formulas for synthesizing chemical weapons, like VX nerve agents, literally one of the most toxic synthetic chemicals ever created. When you add autonomous AI agents to the mix, you risk automating that entire threat design process. To anchor just how seriously the scientific community is taking this threat, listen to this quote from Moratanke at the Johns Hopkins Center for Health Security. The generation of functional viral genomes has urgent biosafety and biosecurity implications. This isn't just science fiction fear-mongering, you guys. It's a very sober, real-world assessment of the risks associated with generative AI possessing the power to design whole viral genomes.
Which brings us to a massive dilemma. How do we manage this? How can the scientific community secure these models from bad actors without completely shutting down the vital, life-saving medical research we just talked about? Well, the Nuclear Threat Initiative, or NTI, has proposed a really smart solution called the Tiered Access Framework. It manages access based entirely on the inherent risk level of the biological AI tool. Take a look at this table. For a Level 1 tool with very low risk, developers can provide the full open-source model to anyone. But notice Level 4, the highest risk category. For a Level 4 tool, handing over the full model, the code, the training data, the weights, should only be done for identifiable, legitimate users who have a specific, vetted project.
Now, you might have noticed the framework also mentions an API. An API, or Application Programming Interface, is absolutely critical here. It's an interface that allows users to send questions and get answers from a biological AI tool without ever downloading or accessing the underlying model weights, the core brain of the AI. Think of it like ordering from a restaurant menu without being allowed to just walk into the kitchen. This is vital because it balances equity with security. It allows a researcher in a developing nation to use the tool for legitimate science while keeping the model hosted securely on the developer's servers so nobody can strip away the safety guardrails.
So, what exactly do those built-in guardrails look like? It's a multi-layered defense. Step 1 is sanitizing the training data. For instance, the developers of Evo explicitly excluded eukaryotic viruses, meaning viruses that infect complex organisms like us humans, from the training data, which severely limits its ability to design human pathogens. Step 2 is verifying user legitimacy before granting access to those high-tier tools. Step 3 involves actively monitoring query volumes and patterns through that API to flag any weird or suspicious behavior. And finally, Step 4 is implementing prompt-level defenses, where the AI is literally programmed to refuse to engage with inputs containing hazardous keywords.
Which brings us to the end of our explainer today, leaving us with this really provocative question. How do we balance the race to cure antibiotic resistance with the need to prevent the ultimate biosecurity threat? The AI genome revolution is here, and it is the ultimate double-edged sword. It absolutely has the power to generate life-saving medical advances out of mere code, but only if we can build the robust frameworks necessary to wield that sword responsibly. It's definitely something to think about as this technology evolves faster than we ever thought possible. Thanks for joining me on this explainer, and definitely keep your eyes on this space. It is moving fast.